Gene mutation, a spontaneous experimentally induced alteration in the molecular structure of a gene which modifies the encoded genetic information, thereby producing a new allele. Gene mutation, therefore, differs from genome mutation and chromosomal mutation. Gene mutations are often point mutation arises from the change of a single nucleotide pair of a DNA of a gene. In point mutation, only a single base pair is affected. In a transition, the purine base pair is affected. In a transition, by other purine base, guanine replaced by adenine or vice versa, and the pyrimidine base replaced by the other pyrimidine base, cytosine by thymine or vice versa. In a transversion, purine is replaced by pyrimidine and the pyrimidine replaced by a purine. In both cases, the number of nucleotides residues remains unchanged, but part of the sequence affecting a single triplet is altered. Deletion or insertion of the nucleotide within a gene results in a frameshift mutation. In a frameshift mutation, transcription still commences at the same site on the DNA and proceeds unchanged to the point where nucleotides have been inserted or deleted. Thus, the number and sequence of the nucleotides has changed. These changes are preserved during transcription and translation so that the final translation is altered. For example, mRNA sequence UUU, CUC, CCA, GAG is translated into the peptide chain flu, pro-glue. Deletion of a single nucleotide, namely the first U produces the sequence UUC, UCC, CAG, GAG, which would be translated as glue. The point mutation that changes the codon for one amino acid into the codon for a different amino acid is called a missense mutation. For the new codon in the termination codon, the change is known as nonsense mutation. Since the genetic code contains only 3 termination codons, most single base replacements result in missense rather than nonsense. Many codons are synonymous that the point mutation may have no effect on the encoded amino acid. Such mutation, namely a change from CUU to CUG, both of which go for leucine, are known as silent mutation. A silent mutation is different from a neutral mutation, which does go an alteration of a translation, but the alteration does not prevent from birth its normal biological function.